Hello, I'm Michelle Taylor, Editor-in-Chief of Laboratory Equipment. Welcome, and thank you for joining us for a 3-minute nucleic acid sample preparation for PCR, qPCR, and sequencing. Today's webinar is sponsored by Cole-Parmer and presented by Laboratory Equipment. Before we begin, I'd like to quickly explain how you can get involved with this webinar. A question and answer session will follow our presenter. At any time during the webinar, you can type a question into the panel through the presentation console. The speakers will answer as many questions as time permits at the end. So today you will hear from 2 speakers. One is Paul Reeve. Paul has worked extensively in the in vitro diagnostic industry for years. He is currently the Molecular Diagnostics Development Manager at Arcis Biotechnology. The company has launched 4 innovative sample prep products recently for research use and diagnostic procedures. Prior to Arcis, Paul worked with both QIAGEN and Epistem on high-profile PCR-based projects. Another speaker for today is Andrew Burney, who gained a PhD in veterinary parasitology in 2008 from the University of Glasgow. He is currently the Global Life Sciences Product Manager for Cole-Parmer. Following some time in research, Andrew moved to that commercial role within the molecular biology sector, and he has worked mainly within companies who specialize in PCR and related technologies. And now, Andrew is going to kick off today's webinar.
Okay, thanks for that introduction, Michelle. So thank you also to everyone who's joined us on this webinar today when we're going to talk about our sample prep technology and its applications in PCR and qPCR as well as directly into sequencing. So before we get properly into Paul's slightly more technical aspects of the presentation, what we're going to do is kind of set a little bit of the groundwork and a little bit of the history that no doubt everyone on the line is more than familiar with. But just as a recap, basically classical methods for doing your sample preparations and your sample extracts fall into one of the 3 sorts of camps. You have a magnetic bead-based system, everyone's favourite, the kind of silica column style, and then your kind of slightly more old school, slightly simplified means of just doing reagents only. Magnetic bead-based systems are basically you have a magnetic bead with a coating on it. That coating will adhere to DNA or will attract nucleotides towards it, and then you, through a means of pulling the beads to the side, you'll exchange the buffers that those beads are in and wash the DNA. You do get as a yield, or as a return rather, you get a high purity. It's a very scalable system obviously because you can do really large volumes, or you can do plates, you can do automation, whatever it is you want to do. There are high binding capacities to those beads, but there are downsides that you still require to add some lysis, and it's a rather expensive system to first get into. Silica columns, everybody who's worked in the lab will have used thousands of these, no doubt, which is basically your spin prep systems. They're cheap, they're a very widely adopted protocol, still requires your lysis, but the thing that I used to find in the lab anyway was the time spent at centrifuges and smashing caps off of tubes and whatnot, which used to irk me somewhat. It was a rather inflexible method, so you're kind of confirmed to, for DNA you have to use this style, for RNA you have to use another style, for blood something, fluid something else, tissue something else altogether. So it's rather inflexible, but it's probably the most common system out there. Reagents based, this is your proteinase K sort of style where you would take your sample, digest it up at temperature, heat and activate it, then do some sort of cleanup, usually like some sort of phenylchloroform. It can take quite a while, and the protocols can take a couple of days to basically do the whole thing, depending how arduous the task is or how particular they are about getting it done. But those are your classic sorts of methods. So you've got magnetic beads, silica columns, and reagent-based. However, that's nothing at all like the system that we're going to talk about today. The Arcis product is unlike any of those. It's totally unique and totally novel. Basically, you have one kit, which is total DNA, total RNA extract in 3 minutes, all at ambient temperature, no heating, no cooling, no centrifugation required at all. You simply follow the 2-step process, which is literally sample goes into into the first tube. You incubate it, or you leave it in there at ambient temperature for 3 minutes, transfer an aliquot of that across into tube 2, and then from tube 2, your DNA and your RNA are ready to be used directly into your downstream application. So as an example, let's say from the picture that you can see on your screen, you've got in the addition of, say, blood into tube 1, which is the lysis stage. So you're adding, say, 30 microliters of blood into tube 1, incubate it for 3 minutes, transfer 10 microliters of that across into tube 2, and from there, that's it, ready to go, completely downstream applications orientated for that. Sample types that have been used so far, and this list is by no means everything that's been tested. There's certainly some interesting things that Paul, no doubt, could tell you stories about later on. But sample types such as most human biological fluids, they've all been tested. Hair follicles, bacteria, gram-positive, gram-negative, virals, parasites like Plasmodium, edible oils, which is a rather tricky thing to extract from because of the high fat content, obviously. And then recently, the guys have done some work on tissue with cow ear, which I believe was a shaved cow ear at this time rather than an unshaved one. However, those sample types have all been tested, and they're all just a small amount into tube 1, 3 minutes, then into tube 2. Downstream applications that have been tested following those have been PCR, both endpoint and real-time, isothermal amplification, Sanger sequencing, pyrosequencing, next-gen sequencing, which Paul's going to discuss a little bit later on, as well as genetic profiling. But really, it's unlike anything that I've seen on the market before. It's no instrumentation required, total extract from just this simple 3-step process with 2 tubes. Really is a phenomenal technology. If we move on to the next slide now, we're basically a little bit more involved with what's actually happening in both tubes. So once you take your sample, you add it to tube 1, what's actually happening in tube 1? Well, in tube 1, everything is becoming lysed. All your membranes are being degraded and your nucleotides are being released. At this point, however, your sample cannot be used because what happens is your nucleotides are aggregated together and they're not in an accessible form to enzymes, which then, for those of you who are thinking about it, is actually a rather useful thing because that means that they're not subject to being degraded. So your nucleotides are actually being protected by being associated with the contents of tube 1. So for people doing field work or field collection assays, potentially you could draw your sample of blood, take your sample, whatever that be, sputum, saliva, buccal swabs, whatever, put that into tube 1, screw the cap on in tube 1, and then that is your DNA and your RNA have been extracted and are stabilized and stored in tube 1 before you ship it back to the lab. You would then transfer that into tube 2, and then from tube 2, you're ready to go downstream. Because in tube 2, your DNA and your RNA or your nucleotides become relaxed, and it's now accessible for your downstream application. Moved on to a slide here where you should be able to see in front of you a lipid bilayer, a cell membrane, and a large circled molecule in the middle. This is where the magic happens in the sample prep kit that we're discussing. Basically what we have here is a molecule which disrupts cellular membranes. So any lipid bilayer which is in there will be destroyed and disrupted. The nucleotides which are then released are then attached to a functionalized ingredient within tube 1, and that's where they become aggregated together and bound together before going into tube 2. And what you'll see here are a series of high resolution or high magnification microscopy on the various stages of relaxation of your nucleotides as they've went in. From that first column on the left would be as they've initially went into tube 2 from tube 1, where they've all been compacted and stored and stabilized. And then as they're in tube 2, they become relaxed. This happens very, very quickly within tube 2. It's not like you need any further incubation or anything like that. Simply just once you aliquot into tube 2, this is what happens, and I'm just showing you that by visualization. Once you get to this relaxed state, obviously that's then accessible to enzyme, be those degradation enzymes like RNAs or DNAs, if there were any in there, or if you were doing like an RT step as well. So if you were taking your RNA extract, doing an RT step to make it DNA to stabilize it before PCR, then you would do that at this time as well. Once your sample is ready and in this stage here of tube 2, you would treat it as any extracted nucleotide. So if it was DNA, you could store it at -20 +4, however long you wanted to do that. Or if it was RNA, obviously you'd put it straight into a -80 from here. So that's the kind of brief introduction to the kit. It really is that simple. It's content into tube 1, 3 minutes in tube 1, transfer a small aliquot across to tube 2, and from tube 2, you're straight downstream into whatever application it is that you want to do. And the main point about this as well is the fact that there's no centrifugation, there's no heat, there's no washes, there's no waiting for ages for specific things to be heat inactivated or whatnot. It's simply just into tube 1, transfer to tube 2, and that is it. Now what I'm going to do is we'll pass over to Paul Reeve, and Paul will talk us through some more specific applications of the sample prep. So over to you, Paul.
Thanks, Andrew. Hi guys, this is Paul Reeves here from Arcis Biotechnology. We're a sample prep company, like Andrew said. So I'd like to start off with some of the fundamental questions about sample prep and about biological cell walls and cell membranes, and some of the questions you might ask yourself as a researcher when you're faced with a sample that you need to process. So often it helps to decide what you would like to do with the RNA or the DNA and work backwards from there. For example, if sensitivity is a critical factor for your experiments and you're looking for a rare target in a background of, for example, wild-type DNA, then you may traditionally think you need purity, you need isolation, you may even need concentration of those nucleic acids. However, sometimes when sensitivity is not required, maybe you're doing a quick screen, a quick yes or no, then good enough is good enough. So you would be looking for a de-skilled process, a high throughput sample prep system. And if I move on to the next slide, fundamentally, sample prep complexity increases as the durability of the organism you're trying to process becomes more challenging. And this is kind of stating the obvious, but I think it leads us on into the fundamentals of the Arcis kit and some of the advantages that we provide over traditional solutions. So, like I said, as a general rule, the more durable the cell wall or cell membrane that encapsulates the nucleic acid, the more complex the sample prep procedure becomes. On the easy side of things, we've got lipid bilayers, which Andrew showed you on that cartoon slide. Very easy to lyse, found in things like epithelial cells. These are really fragile. They can be disrupted by the most gentle of even chemical interventions, often simply by osmotic pressure alone. So these cells will burst in water. They're so fragile that the osmotic lysis can open them up. You don't need very much sample prep for these easy targets. As we move along the complexity scale, you can encounter the bacterial cell wall, the main component of which, the most important component of which, is peptidoglycan. So peptidoglycan, for those who don't know, is a polymer of amino acids and sugar. It makes up the cell walls of bacteria. So it's this layer that differentiates these microorganisms from the simple epithelial cells. The peptidoglycan protects the inside of the bacterium, the relatively salty inside of a bacterium, from the osmotic pressure of the more dilute surrounding fluid. And it's for this reason, it's the peptidoglycan layer that stops bacteria simply bursting open in water. So for bacteria, you need a slightly more complex approach. Traditionally, chemical methods may be still effective. Surfactants like SDS can easily break open E. coli, for example. But the thicker the peptidoglycan layer, the more complex the sample process becomes. And as you may know, it's the thickness of the peptidoglycan layer that we use traditionally to divide bacteria into gram-positives or gram-negatives. The thickened peptidoglycan layer in the gram-positive cells allows them to retain the gram stain, hence the name gram-positive, whereas the thin layer in gram-negative cells, they cannot prevent the gram stain from leaching out. So under the microscope, you can very quickly tell if you've got a gram-positive or a gram-negative. So it follows then that gram-positives with the thicker peptidoglycan layer, things like staphylococcus or streptococcus, these often require a more complex sample prep procedure than a gram-negative like E. coli or Klebsiella. At the higher end of the complexity scale, we find organisms that are encapsulated with complex polysaccharides such as chitin and cellulose. For example, spores. These present a real challenge to conventional sample prep methods. And for spores, the most complex sample prep procedures are recommended. So these may be combinations of things, so you may need to heat, degrade by enzymes. Combination approach, the most complex approach, is often used for something like spores. Chitin, which is a principal component of a fungal cell wall, presents a similar challenge. Chitin is structurally similar to cellulose. Cellulose and chitin are both traditionally present real challenges to sample prep. And the most complex workflows with several lysis and washing steps will be used for these samples. However, as the complexity of the sample prep process increases, by definition, the amount of energy that you need to input into the sample prep system increases alongside. So, the basic tools at our disposal, I'm talking fundamentals here of sample prep, are listed on the slide: chemical, enzymatic, thermal, and mechanical methods of lysis. Each require increasing amounts of energy, and each comes with its own set of pros and cons. So the more aggressive the sample prep process, the more opportunity for sample loss. And the more aggressive the process, the more opportunity for nucleic acid degradation. For example, you could take a sample and simply pour household bleach onto your sample. You will lyse the cells in most cases, but you will also destroy the information that's contained within the cell, and then you can't do anything useful with the sample at all. So a trade-off must be found between lysis efficiency and protection of the important biomolecules found within. And often the trade-off must be found empirically, experimentally, and by the accumulation of evidence. And because this is biology, samples come in all shapes and sizes, and there's very rarely a one-size-fits-all protocol. However, at Arcis, we've taken steps, indeed, to provide a one-size-fits-all protocol. It's a quick, like Andrew says, de-skilled process, very simple, and as the following slides will show, the kit can process the widest range of biological samples with what we think is unparalleled speed compared to anything on the market right now. Our standard protocol is chemical in nature. Like Andrew says, there's no need for instrumentation in most cases. You simply add your sample to reagent 1 and then wash with reagent 2, and that's all. So not only is it a relatively gentle system, gentle but effective, as you'll see, it doesn't damage nucleic acids. In fact, the opposite, it actively protects the nucleic acid populations, and that's DNA and RNA. So in our development labs and in collaboration with our customers, we've optimized a set of protocols for common molecular detection techniques, for standard laboratory techniques such as PCR, qPCR, sequencing of all flavors, and strange things like isothermal amplifications, genetic profiling. I'll drill down into the data in the slides coming up, and you'll be able to get a flavor for kind of the breadth and the scope of the kit, and hopefully you'll get some ideas of how you might use the kit in your research. So if I can begin drilling down into some data and talk about sample prep for PCR. So at Arcis, most of our development studies have focused on real-time PCR, real-time PCR being obviously one of the most widely used molecular techniques, heavily used in research, academia, government laboratories across the board. And as the next slide will show you, the Arcis kit is platform and chemistry agnostic. So it doesn't really matter if you're using a rotogene, a light cycler, ABI, step 1. we get consistent results across the board. We've got extensive data actually on other PCR machines, miniaturized, tabletop, palm-top PCR machines, and I'll show you some of that data later on. But as you may know, different commercial PCR master mixers contain slightly different chemistries. They're all based on the same fundamental principles, but different platforms may differ in ramp rates, heating, cooling, or different for detection channels and dyes. But we've found, and hopefully you can see on the slide here, a rock-solid consistency between the most popular commercial qPCR master mixers on the market today. So I'm confident in saying that the chemistry is platform agnostic, and our kit is in fact master mix chemistry agnostic as well. So if I can talk about what we see as a relatively easy sample to deal with, traditionally viewed as something that presents kind of a problem to researchers in terms of detection, and that's whole blood. So with the Arcis kit, you can go from whole blood to PCR in about 3 minutes. Using the very simple process that Andrew outlined earlier, you simply add an amount of blood to reagent 1, usually in a 1:5 ratio. Then you leave that at ambient temperature. This will perform a controlled lysis of the cells. So it doesn't just blast the cells open, it performs a far more controlled lysis of the sample. And then you wash, and you're straight into PCR. Interestingly, and if you try the kit with blood, you will notice this with your own eyes, the processed blood sample can still have a reddish-brown tinge to it. And this makes some researchers run a mile. They're like, how can I possibly detect my products if the starting sample is red or it contains some kind of color that's going to interfere with detection? But that's where the clever chemistry in reagent 2 comes in. Reagent 2 is a potent scavenger of PCR inhibitors, including heme. So although you will see heme, a red tinge to your sample, don't worry about it. Proceed to your downstream testing protocol and see what results you get. For us, whole blood is a sweet spot. We perform qPCR, PCR sequencing with whole blood, and the chemistry of reagent 1 and reagent 2 mean that the heme will no longer interfere with your polymerase, and you can get good data in an ultra-rapid turnaround time. We've tested this very simple protocol with probe-based PCRs, qPCRs, and dye-based qPCR. So those probes, I'm talking about TACMAN, I'm talking about some of the hybridization probes like molecular beacons. We've got internal data on most detection technologies for qPCR. So that's whole blood. But let's move on to bacteria. And in this case, the data on the slide come from Klebsiella pneumoniae, which is a gram-negative bacterium. And if you follow the same protocol, in which you add your sample to reagent 1, allow that to lyse at room temperature for 60 seconds, and then proceed with the protocol, you can go from bacteria to amplifiable DNA or RNA in a couple of minutes, and then you're ready for PCR. So as I kind of hinted on the earlier slide, reagent 1 performs a controlled lysis of cells. So it doesn't just blow the cells wide open and completely disrupt the membrane. Actually, molecularly punches holes in cell walls. And in fact, in the right conditions, you can use reagent 1 as a transfection reagent to make cells competent. So that might give you some insight into the controlled nature of the lysis step in this kit. It's not simply a detergent going in and wiping out the cell membrane. It's a more complex and it's a more controlled process than that. So if I move on from gram-negative bacteria into an example of a gram-positive, which here is shown Staphylococcus aureus. So once again, we took cultured staph, or we picked a colony of staph and diluted that into reagent 1. And the exact same process happens again. You go from gram-negative bacteria to PCR in a couple of minutes. Now, don't forget that you are carrying over debris from the lysis reagent, reagent 1, into reagent 2. and you are in fact carrying over some cellular debris into your PCR reaction. But like I hinted earlier, reagent 2 is the important part of this process. It scavenges those inhibitory factors and simply takes them away from your qPCR reaction. So in some cases, you might take a hit on sensitivity, but you gain in simplicity. So the turnaround time goes from, let's say, 45 minutes for a traditional spin column kit to more like 4 minutes, less than 4 minutes with our system. And the performance is comparable between the two. So that was bacteria. I'd like to show you now some data on parasites. We have a clinical development laboratory that has access to many tropical diseases, lots of real patient samples from infected blood, and we thought we would try a quick test on Plasmodium falciparum, the causative agent of malaria. So we're now processing a parasite in blood. Same principle. You add your patient sample to reagent 1, follow the standard protocol, and you're ready to go into PCR. This was an assay that we actually developed in-house to prove a concept. So this was not a commercial Plasmodium assay. This was an in-house laboratory developed test in our clinical laboratory. And we were simultaneously detecting RNA and DNA from the Plasmodium. So by the primer design, we were actually able to detect both populations of biomolecules. And the effect of this, as the next slide will show you, was a massive boost in sensitivity. So the graph shows you a cohort of 13 patients, malaria-positive patients. These are real patient samples. And the blue dot is the gold standard clinically validated kit. So this is the gold standard kit that they use for malaria diagnostics in this particular hospital. We asked them to do a side-by-side with samples processed with the Arcis kit, and as you can see, on average we gained about 10 CTs improvement in sensitivity. So this showed that not only can we go from rapid sample prep, we can detect RNA and we can detect DNA. And although our process is dilutive, the inherent Arcis process is dilutive, you can overcome any kind of dilutive effects by primer design and by exploiting the fact that all of the nucleic acids are captured. Nothing is lost with the Arcis reagents. So it's not like a spin column where you lose with every wash, or a magnetic bead extraction where your efficiency depends on the chemistry of the tube. There are no losses with the Arcis kit. Everything that you lyse and drop into solution is captured. There is a real robustness to the kit. Everything is scalable. So although the standard protocol and most of our initial development studies used 30 microliters of starting sample, and this was chosen because that kind of lines up with what you would expect from a finger prick of blood. So you really do not have to do 30 microliters per sample. We've gone all the way down to less than 1. We've gone all the way up into milliliters. Everything is scalable. So no matter what sample you're faced with, no matter what the volume, you can use the kit. The whole process is indeed scalable. And in the right conditions, as the slide shows here, you can drop the amount of reagent 1. So we were faced with a challenge from a customer who wanted to make a microfluidic device, for example. Everything needed to be miniaturized. So we did the study, and indeed, everything can be miniaturized without any loss of performance. So what the message from this slide is, sample input, scalable, and the processing protocol, the amount of reagent 1 and reagent 2 that you need to use, is also scalable. So this can give you the confidence that no matter what sample size you're working with, we've probably got a protocol that will work with your workflow. And to reinforce this point, if I move on to the next slide, which was taken from an analytical sensitivity study from human blood, we diluted the whole human blood sample down to 0.2 microliters, and we still got detectable amplifications from a single copy human housekeeping gene. So this is whole blood. Again, this is a sample that looks red when you load it into your PCR plate, but we were able to detect as low as 0.2 microliters of whole blood in a standard qPCR instrument. So hopefully this reinforces to you that you can go low on sample input, you can go high. We generally have a sample prep protocol that can suit your needs. So like Andrew said, not only do we release nucleic acids, we protect the nucleic acids. The chemistry in reagent 1 means that as soon as DNA or RNA molecules come into contact with the reagents, they form thermodynamically stable clumps. They group together, they clump up, and they are therefore physically inaccessible to specific degradative enzymes. So this opens up opportunities to use the kit as a sample collection and preservation system. For example, you can add any biofluid or any sample, be it on a swab, on tissue, on dried card, dried blood, anything. You can add your biological sample to reagent 1 and it will be preserved at room temperature for at least 30 days. Now we've got data which extends out way beyond 30 days, but right now we're very confident that RNA and DNA molecules, all of the population is completely protected for 30 days on your bench at room temperature. So this opens up opportunities for maybe people who are collecting samples and they need to batch them before they process them. You can keep them on the bench in reagent 1. Or for people who may be collecting samples in strange locations, maybe in-field, agricultural, aquaculture type applications. So if you collect samples in strange locations, you can take some reagent 1 with you, drop the sample into reagent 1, and it will be stabilized. Now I need to point out that the reagent 1 is lytic. So it's not like a sample collection system like RNA later or PAXgene, which protects cells. They prevent lysis of cells. We don't do that. We actively lyse the cells, but we protect the nucleic acids. So this is a very different take on sample collection and preservation because lysis will happen. And because lysis happens, the samples can be disinfected. So if you're faced with collecting samples of an infectious nature, you can be confident that they are stable in reagent 1 and they're also disinfected in reagent 1. So this can kind of solve several problems in one. And by the time you get back to the laboratory with your sample in reagent 1, most of the sample processing is done. All you need to do is go into reagent 2 and then you're into PCR. So you can save yourself a lot of resource just by using the Arcis kit as a sample collection and stabilization system. So I'd like to show you some data which exemplifies the phenomena that I've just described. So this graph, which is more or less 2 straight lines, and each straight line is a different human saliva donor. So this was basically scientists in our lab, saliva, 30 microliters of saliva into reagent 1, and then we left it either on the bench. So lab temperature on the bench is about 18 to 22 degrees C. Or we place these samples into an incubator at 40 degrees C for accelerated stability testing. So even at 40 degrees C, and this is and counting, we're still seeing rock solid stability for 97 days. I think we're over 100 days now. This slide is a couple of days old. But we will keep testing until the sample falls over. But for now, we are basically up to 100 days at 40 degrees C, and we are seeing no loss of performance. So this is a good box for us to tick that we really do preserve the samples. And watch this space. We keep testing and we keep testing. 100 days at 40 degrees C. Somebody more intelligent than me can do the Arrhenius equation and let me know what that equates to for real-time stability, but it's a long time. And it's probably long enough to suit most people's needs. So that was human saliva. Here are some data from human blood, again from 2 donors in our laboratory, and the same protocol was followed, in which we took a blood sample into reagent 1, either on the bench or in the 40 degrees C incubator. And in this case, we can see some drift in the CT value on the blue lines of the graph. They are the 40 degrees C accelerated stored samples, but the orange and the red traces are rock solid. So we're seeing some degradation in this experiment. I mean, there are error bars on all of these time points, by the way, these averages. But I think we can safely say that compared to an unprotected sample, we are seeing really, really strong preservation of the nucleic acid populations in these samples. So that was whole human blood. This sample is the same process with a gram-negative Klebsiella and a gram-positive bacterium staph aureus. Again, the take-home message is it's a stabilized system. There's possibly even some benefit observed in this experiment to the agitation of the samples as they go from the incubator onto the bench. Over time, we're seeing an increase in lysis efficiency sometimes from day 1 compared to day 28. For the most durable organisms, everything is lysed within a few minutes. And this is experimental data. Remember, there are error bars on all of these time points. But the take-home message is you can store your microbiological samples in reagent 1, and they will be stabilized. So I think this slide kind of brings me to the end of PCR data. Most of our development studies are conducted on Roche light cycle instruments or ABI step 1s. And like I said earlier, please try the kit on whatever chemistry you're using, whatever platform you're using, and we're confident that you'll get good performance. But if I can move on slightly from the qPCR data and talk about sample prep for sequencing. So in our development studies, we've tried the following. We've tried Sanger sequencing, we've tried Pyro, and we've taken early steps into sample prep for next generation sequencing. So if I begin with sample prep for Pyro sequencing. So this slide, the orange boxes show you the traditional sample prep and library prep that goes into experimental setup for Pyro sequencing. It's a long and arduous, laborious process. It's a very complex workflow with washing, with extraction, with magnetic beads. In fact, there's a disclaimer here. One of our scientific team actually worked up the chemistry, the PyroMark chemistry, which was later acquired by QIAGEN. So testing Pyro sequencing was an obvious next step for us. Once we'd investigated the molecular phenomena of PCR, qPCR, we've moved into Pyro sequencing. So what the Arcis kit does in this workflow is dramatically reduces the nucleic extraction time. So by no means is it a given that you can go rapidly from cells to DNA and then into library prep. Traditionally, you need cleanup. You need extensive cleanup. But as the next slide will hopefully show to you guys, we've gone from samples. In the case of the next slide, it's clinical samples from liver cirrhosis patients straight into library prep. So we reduced that whole nucleic acid extraction time from 45, 60 minutes down to 3 minutes. And this is a study on this slide that we published a couple of years ago now. We presented it as a poster in the International Liver Congress in 2015. But basically, our development team in Spain, they took a fairly large cohort of patient samples, 151 in this case, and they were all rapidly processed with the Arcis kit. And this was followed by sequencing analysis, and in this study it was both Sanger sequencing and the more sensitive Pyro sequencing. And in fact, we got very good sequencing data for all cases. So 151 out of 151, we got good yields, good quality, and we were able to go from cells to library prep in 3 minutes. So these data here, it's an example of kind of a patient stratification application. The thought behind this study was to identify SNPs in the genome and a haplotype of SNPs that was prognostic for progression from liver fibrosis to liver cancer. So we had a really useful biobank of samples that were ready to be processed, And this was our initial steps into sequencing. So this is Sanger and pyrosequencing data. And we were pleased with the result. We processed a large number of samples very quickly. And this also highlights another feature of the kit, really, because it's a liquid system. It's suitable for liquid handling robots. So you can preload a plate with, let's say, 96 wells of reagent 1, and you can pipette in 96 blood samples, and they can be transferred by the robot into another plate loaded with reagent 2. So you can achieve real high throughput with our kit, and it's fully compatible with liquid handling robots. So if we can stay on sequencing for a while, we moved on from pyro into what is basically massively parallel pyrosequencing, the Roche 454. In this study, it was the GS Junior platform. Still extensively used in clinical laboratories, definitely still used in our development laboratory. And this is more of a work in progress, but we do have solid data, which I'll show you next. And once again, just like the pyrosequencing workflow, we were able to reduce the upfront nucleic acid extraction time. So we went from human blood into library prep in a couple of minutes. The Arcis DNA, although this is something the Arcis DNA kit will fragment DNA. So this is something that we're actively looking into. And in the case of the next-gen sequencing workflow that I show you here, the fragmentation of the DNA is an inherent part of the workflow anyway. So the fragmentation of the DNA by the chemicals in reagent 1 was an advantageous step for this workflow. We dramatically reduced upfront turnaround time, and we were able to, as this next slide will show you, we got a good result basically looking at the human hRNase P gene. So we followed the standard workflow. So it's nucleic extraction, fragmentation of DNA, adapter ligation, and then the complexity of sequencing in the Roche 454. But this, for us, was a good box to tick. We've gone from our very rapid nucleic acid extraction in front of a next-gen sequencing platform. Now, I know it's 454. I know it's Roche 454. It's still extensively used in clinical applications. But we've also, in collaboration with other groups, we've got very nice early data on Ion Torrent and Illumina MiSeq. So if you're conducting next-gen sequencing experiments in your laboratory, please get in touch with us. Although this is a work in development, we've got great data and we've got really, really promising protocols, and we can get that turnaround time way down if you use our kit. So this is where we stand for next-gen sequencing. It's as I said, it's a work in progress. We'll be able to share much more exciting data when we can go public with it. We've got some posters and publications in manuscript, in process of writing up right now. But when we go public with those, please be sure to check them out. It's a major step forward, we think, for sample prep for next-gen sequencing. So I'd like to move on from sequencing and talk about some more specialist applications for genetic testing, particularly forensics type applications, human identification workflows, or methods where your sample collection is slightly non-standard. So maybe swabs or biopsy punches or veterinary samples such as tissue, animal tissue. And the following slide is data from blood samples which were dried. So this is dried blood swabbed up from the bench. So it's simply air-dried blood dropped onto the bench and picked up by a wet swab, dropped into reagent 1. Or it was dried blood on these FTA Whatman type cards. So this is a very common collection technique. We had blood samples dropped onto the card. We used a 1 millimeter biopsy punch to take a disc from the card once it was completely dry, and we dropped the biopsy punch disc into reagent 1. Follow the standard 3-minute process. And sure enough, we get quite nice results. We get amplifiable, detectable DNA from this slightly more unusual collection technique. So if you're doing any kind of forensics type application, or you're thinking about developing a protocol for these commonly used human identification sample collection types, again, we hopefully have a protocol that can suit your needs. Staying on the subject of human identification sample collection types, we also tried buccal swabs. Now we've got an extensive set of data on buccal swabs. We've got stability data, and we've got extraction efficiency and yield data. But this was one of our first experiments in our laboratory, where we took a buccal swab from a couple of the scientists in the lab. You simply drop the swab into reagent 1, mix, and that could be squeeze the swab, swirl the swab, just anything to try and liberate the nucleic acid from the matrix of the swab, and then proceed washing and into PCR. So once again, these are commonly used forensics type sample collection workflows, and this led us on to a full-on pilot study into a forensics type human identification workflow. So in this study, we compared the Arcis kit with a standard accredited forensics workflow. Traditionally, in this workflow, in this accredited workflow, it takes between 3 and 4 hours to get nucleic acid from the sample. So we compared this 240-minute system with our 3-minute system. And as the data show, we did a small cohort of samples. We got DNA of a good quantity and good quality. Indeed, we ran from start to finish of the workflow. And we got full profiles for 6 of the samples. We got partial profiles for 2 of the samples. When you inspect those 2 partial profiles by eye, you can see that there is sufficient information for full identification. And indeed, the results of this test, this quick test, the results were actually good enough to go onto the UK National DNA Database. Of course, we didn't enter these results into the database. It was just a trial run. But it showed us that we can go in minutes from a forensics type sample to something which gives us the quality and quantity required for this lockdown accredited workflow. So this was just a nice box for us to tick. For anybody who's doing human identification studies, we thought this is a good showcase for our technology. I'd like to speak briefly about point of care detection. Obviously, because our kit is stable at ambient temperatures, I mean this is UK temperatures and elevated temperatures, you can take your sample collection into the field. So if anybody on the line here is doing in-field collection or pen side or pond side, you might be doing agricultural testing or aquaculture testing. You basically pick up your sample, drop it into reagent 1. You don't need any cold chain storage. Everything is stable, and you can batch your samples, or you can collect your samples and take them into your downstream testing with safe in the knowledge that your sample will be preserved. You don't need any power for this process. You don't need any instrumentation, and that's what makes it almost uniquely suitable for point-of-care detection. Furthermore, as the following slide will show you, we can actually lyophilize these reagents. So this completely eliminates cold chain logistics. It makes it truly point-of-care applicable, and it comes with an increase in stability and shelf life. And as the slide shows you here, we compared the performance of our liquid kit with our lyophilized kit, and we got exactly the same performances. So the 2 graphs on the screen there show you 2 traces which basically sit right on top of each other. So that's another option. If you've got any studies that you may think require lyophilized reagents, you can get in touch with us, and we can work together. And if I can stay on the subject of point of care detections, I've got some example data here of a comparison between our kit and a commercial point of care sample prep kit, which uses magnetic beads, and the complexity is increased So there's a fair bit of hands-on time, and there's washing and elution steps, as you traditionally get with magnetic bead extractions. And these are actually the last slides I'm going to show you on the presentation here, but it's a side-by-side comparison of our kit with a commercial point-of-care system. The red trace on the graph is the commercial magnetic bead system. The blue trace is us. And for whole blood, we got almost exactly the same performance as this 20-minute relatively complex system. For plasma, same story. We got the same. And I'm just going to flick through these slides now. Serum, again, performance was the same between the two systems. And finally, urine. So we got really nice results with urine, and that opens up lots of opportunities. You know, urine is a fantastic liquid biopsy system, increasingly used in diagnosis of human illness, of Mendelian diseases, and indeed of infectious diseases. So we're confident that we can use any human biofluid. We can use them in a laboratory setting. We can use them in a point-of-care setting. So hopefully I've shown you enough data here to kind of whet your appetite, and if you have any questions, please fire away, and we'll move on to the Q&A section soon. But thank you for your attention so far. We do have a summary and FAQ slide, which I'm not going to read to you, don't worry, but it probably answers some of the questions that you might have about our kit. And it talks about yield, it talks about scope of the kit, stability of the reagents, and just other questions that you may have. So I think that's where I'm going to leave the presentation. Thanks for listening, and I'll leave you with a summary slide there, and we can, I think, move on to questions.
Yes, thanks, Paul. That was some great information. Audience, we do have, about 10 minutes or so for questions. So we can jump right in. There's actually an incredible amount of questions. So let's get the first one, so we can try to get to as many as possible. The first question we have is for you, Paul, so you don't get a break from talking. Is it possible to quantify DNA post-lysis by fluorometry for next-generation sequencing applications?
So what we recommend is, unfortunately, and nobody likes to hear this, we recommend standard curve qPCR for quantification. Although a system, a DNA binding fluorescent dye type quantification system, let's say qubit, you do get, suitable results. Whereas for something like nanodrop, you do not get good results with this kit. You just get noise with the nanodrop, so it's completely unsuitable for spectrophotomic quantification. But to be on the safe side, and this is less than ideal, but you need to do standard curve qPCR for yield determination. That's really is the gold standard with this kit.
Okay, great, thanks. Now let's get Andrew back into the fold. This question's for you. How do you pipette cow ear or hair follicle? That's an interesting one.
Yeah, I knew that was going to come back and get me. Obviously that's not pipettable. So what you would do is take a small punch of that, put that into your sample, and then you could use like a little, if you wanted to, you could use like a little in-tube homogenizer to kind of disrupt that a little bit. Yeah, I knew that was going to come back on me, that one.
Well, thank you. Now we know how to, so that's always a good thing in case anyone needs to do that.
Yeah.
All right, so Paul, now we're going to move back over to you again. In the sample prep procedure you guys were talking about, does tube 1 contain any dangerous materials?
No, it doesn't. It's a really boring safety data sheet. It's non-hazardous. It's very safe.
Okay.
Very boring.
Well, probably not too bad then. Right, still talking about the tubes. What is the volume of tube 2 compared to tube 1? What is the dilution factor?
That's a very good question, and dilution is an inherent part of our protocol. So the ratio from tube 1 to tube 2 is 1 to 4. So there you go. You've got a 1 in 5 dilution. You go from, let's say, 10 microliters tube 1 into 40 microliters of tube 2. That's our standard recommended protocol. But we realize that this, will impact sensitivity. So for protocols where you really do need sensitivity, that ratio can be completely altered. You can go 1:1. So you reduce the dilution factor considerably. And for people for whom 1:1 is still not good enough, we can do lyophilized reagents, which will completely eliminate dilution effect. But that's a non-standard product, obviously. The take-home message is the ratio is 1:4, but that can be scaled and changed to suit your needs in terms of sensitivity and yield.
Okay, great. And now, is there a difference if a researcher wants to extract DNA versus RNA?
Nope, is the quick answer. No, we have to, all nucleic acids, all populations of DNA and RNA, it's the same procedure.
Okay, sounds good. Here's a specific question regarding agriculture. Are you planning on testing your kit on plant tissue?
Yes, the answer is yes. And I mean, for lots of our collaborations, we're tied up in confidentiality.
Okay, well then really quick on that. Will a high phenol content in wheat interfere with this extraction kit?
I can confidently say no. Nope. There's so
Great.
much scavenging activity going on with reagent 2 that, you can proceed with plant material with confidence.
Okay, perfect. One more for you, Paul. Are there any restrictions due to the size of nucleic acid?
No. I mean, you need to bear in mind that the kit will fragment DNA. So if you're looking for massive reads, for example, if you were using a PacBio next-gen sequencing machine and you needed 30 kb stretches of DNA, this might not be the right tool for the job. But in terms of restriction on nucleic acid length, no, not really. It'll suit 99% of applications.
Okay, great, thank you. And Andrew, we're going to jump back to you for a second here. This question is for you. Is the Arcis malaria kit the same as the Arcis?
Yeah, it's exactly the same kit. There's no change in the recipe or the formula or the protocol at all. It's exactly the same.
Okay. And also, what volumes of reagents are provided in the kits?
It's 50 reactions for the kit, and that's 50 reactions of 150 microliters of tube 1 and 50 reactions of 40 microliters of tube 2.
Okay, thank you.
Yep.
And Paul, back to you quickly while our time is running out. Can researchers use direct saliva without the swab?
They can indeed, and the stability data that I showed in a slide was direct saliva straight into tube 1. So yes, the answer is yes to that one.
Great. Right, and let's do one more question here for Andrew. What is the compatibility of the reagents? If you're using, if you're looking at DNAS, if you only want to look at the RNA.
Oh, okay. So the question's about converting RNA over to DNA. Yeah. Okay, so no problem at all. What you would do is you would do your extract, you would do your standard protocol, take it through into tube 2, and then you could incubate tube 2 with DNAs in there so that you digested away the DNA before you performed your RT step. You would simply do that step in accord with the protocol stipulated for your reverse transcriptase kit. No problem there at all.
Great, thank you. Now, audience, we're actually running out of time. We had so many great questions by you guys. So what's going to happen is Paul and Andrew will receive a spreadsheet of all the questions immediately following this, and they will be able to follow up with you personally on your question if it was not directly addressed in the last 10-15 minutes. But otherwise, thank you so much, everyone, for joining us on our webinar today. Thank you to our speakers, Andrew and Paul, for their expertise, and a special thank you to Cole-Parmer for sponsoring the event. In addition to the questions that they will get back to you on, this webinar has been recorded, so you can always come back and watch it on demand within 24 hours. Have a good day.